This is Sarah Wilkinson from Humber College and the University of Guelph-Humber. In this video, I'm going to provide an overview of oxidation and reduction reactions in ATP production. In a previous video, I covered what enzymes are, we talked about ATP, and the phosphagen system. In this video, I'm going to talk about how energy is harnessed and transferred in the cell. As you recall, fuel we either eat or stored contains energy, and that energy is transferred to the bonds in adenosine triphosphate. That adenosine triphosphate can then be used to perform physiological functions or produce heat. In order to transfer energy, there are many steps involved. And many of those steps involve energy transferred from one chemical bond to another. When we're talking about energy transfer, what we're really talking about is transfer of electrons and of their associated hydrogen. And when we transfer electrons, we usually are going to be transferring them to coenzymes. And these coenzymes that are used are NAD and FAD. So electrons are transferred from carbohydrate, fat, and proteins to molecules of NAD and FAD. And what we're going to be talking about, we're going to give them terms like oxidation and reduction, whether they're gaining electrons or losing electrons. That's what this video is about. So, when we oxidize a substance, such as glucose, what we're talking about is it losing its electrons. It's losing some of its energy. So glucose being oxidized will transfer electrons and hydrogens to a coenzyme, say NAD, and that NAD will receive those electrons and hydrogen and become reduced. So oxidation and reduction are always paired. So one substance is oxidized, glucose, and the other is reduced, NAD. Here we go. We've got a substrate molecule, for example, glucose, and we've got another substrate molecule, NAD. NAD being the coenzyme which is going to receive electrons and hydrogen and become reduced. The substrate here was oxidized because it lost electrons, and the coenzyme NAD was reduced because it gained electrons. So NAD becomes NADH plus H. If we're talking about the other coenzyme, FAD, it becomes FADH2. So oxidized form, NAD or FAD, reduced form, NADH or FADH2. What this is allowing is the transfer of electrons from the substrate to a coenzyme. And that coenzyme then enters the electron transport chain, which we'll talk about in future videos. This slide provides an overall summary of what we're going to be covering in future videos. So if it doesn't make sense at this point, don't worry, it will become clear as the videos go on. What I wanted to point out is that in each step in metabolism, so if we start with a molecule of glucose and convert that to 2 molecules of pyruvate through glycolysis, NADH is produced during that. When we convert pyruvate to acetyl-CoA, NADH is produced there. When acetyl-CoA goes through the Krebs cycle, NADH and FADH2 are produced there. All these reduced coenzymes can then enter the electron transfer chain and transfer electrons and hydrogens, eventually to produce ATP. If we're talking about fat metabolism, when acetyl-CoA is produced, NADH is generated there. And obviously, because we're producing acetyl-CoA, it goes through the Krebs cycle, and FADH2 and NADH are produced there, which can go to the electron transfer chain. The bottom line from this slide is that coenzymes are reduced, i.e. they gain electrons through a series of chemical reactions that transfer energy from carbohydrate, from fat, or from protein to the coenzymes. Those coenzymes then enter the electron transport chain, and that energy is harnessed in the electron transport chain to produce ATP. Upcoming videos will cover how this is done. So we've got one video on anaerobic glycolysis and 3 videos on oxidative phosphorylation. I would encourage you to watch those, and the previous slide will become much clearer.